IVF. It involves extracting eggs from the ovaries, processing them in the laboratory, fertilizing them with a sperm in a dish, culturing them for some days to grow and divide, and then transferring the healthy embryos back into the womb. But how do we know if these embryos are boys or girls? We all started from one cell that divided into 2, then 4, then 8, till it developed into a full-grown baby. Every cell has 23 pairs of chromosomes, 23 from the sperm and 23 from the egg. The sperm cell can have one of 2 different types in the last position, an X chromosome or a Y chromosome. This is what determines the sex of an embryo. If a sperm with an X chromosome fertilizes the egg, it creates a girl. Or if one with a Y chromosome fertilizes the egg, it creates a boy. But since, in some cases, we select the sperms ourselves, can we intentionally create a boy embryo or a girl embryo? Kind of, actually. The X chromosomes are quite larger than the Y chromosomes. So sperms carrying these X chromosomes tend to be slower and heavier than those with the Y chromosomes. So theoretically, we can sort the male sperms from the female sperms. But techniques using these strategies have been found to be very inaccurate. We can look at the larger picture though, the embryos. Looking at the embryos under a microscope won't reveal its chromosome makeup. But the good thing is, we've got many more cells. Since the embryos can continue to divide and heal themselves, we just pick out a few cells and send them to the lab for testing. And cryopreserve the embryos till when the results comes out. This is what we call pre-genetic testing, or PGT-A, because there are different types of genetic testing. PGT-A can reveal an embryo's sex during IVF. But how exactly does this work? Here's the breakdown. PGT-A involves a delicate procedure where a few cells are biopsied from a more developed blastocyst stage embryo. These cells are minuscule, so it requires some serious skills from the embryologist. Once extracted, the cells are then sent to the genetic lab where DNA from the cells are fragmented and copied millions of times using the polymerase chain reaction. Using a next generation sequencer, the copies are then sequenced and analyzed with a software to identify the type of chromosome, that is the sex, and abnormalities in the number or arrangement of the chromosome. So yes, PGT-A can detect other abnormalities such as Down syndrome or Turner syndrome or other chromosomal defects. Actually, that is the ideal use case for it. Sex selection is just an additional perk, and some might say it is wrong ethically. You see, the world of IVF is in a constant fascinating dance between science and human desire. Is IVF wrong? Is cryopreserving embryos wrong? Is selecting embryos based on certain criteria wrong? We've explored the microscopic world of embryos, but the ethical questions surrounding sex selection still remain. What do you think?